This video will be explaining the process of mitosis. But first, what is mitosis? Mitosis is the process a cell undergoes in order to divide into two identical daughter cells, which are the result of the process. For the cell to undergo mitosis, it must first go through interphase so that the cell divides properly. The first step of mitosis is prophase. During this phase, the DNA inside of the nucleus starts to condense, and the sister chromatids join together at the centromere. The nuclei also begins to degrade in order to release the DNA. The last thing that occurs during this phase is that the centrosomes start to move to opposite sides of the cell and the microtubules begin to form. In prometaphase, the nuclear envelope breaks, allowing the microtubules to invade the nuclear area. The chromosomes condense more, and each of the two chromatids have a specialized protein structure, called a kinetochore, attached at the centromere. Some microtubules attach to the kinetochore, becoming kinetochore microtubules, and this pull the chromosomes back and forth until they settle in the center. Other microtubules, the non-kinetochore microtubules, interact with its counterparts from the opposite pole of the spindle.
Metaphase is a stage in the cell where all genetic material is condensing into chromosomes. These chromosomes then become visible. In a eukaryotic cell division, chromosomes are lying on the metaphase plate in the middle of the cell.
Anaphase begins when the duplicated centromeres of each pair of sister chromatids separate, and the now daughter chromosomes begin moving toward opposite poles of the cell due to the action of the spindle. Appears during chromosome movement. At the end of anaphase, a complete set of chromosomes has assembled at each pole of the cell.
Telophase is the final stage in cell division. During telophase, the nuclear envelopes reform from the new nuclei in each half of the dividing cell. The ribosome-producing portions of the nucleus return. As the cell has finished moving the chromosomes, the main parts of the spindle apparatus fall apart. As telophase moves towards completion, the chromosomes are released from their tightly bound structure back into loose chromatin. The division of the cytoplasm begins by late telophase. In animal cells, cytokinesis is demonstrated by the appearance of a cleavage furrow that pinches the cell in two. In plant cells, however, there isn't a cleavage furrow. Cytokinesis begins in telophase, when the vessels from the Golgi apparatus move microtubules to the middle of the cell, where they produce a cell plate. The cell wall materials carried in the vessels collect in the plate, making it grow until it fuses with the plasma membrane along the perimeter of the cell. At the end, each daughter cell has their own plasma membrane, and a new cell wall arises from the remains of the cell plate between the two cells.